Hello everyone, this is Doug, and in this video we're going to take a look at glycolysis. We're going to look at some of the nitty-gritties, talk about general enzyme types, and look at the cofactors and coenzymes that are needed to perform glycolysis. So just as a quick reminder, we're talking about cellular respiration in general, where you have your formula of glucose and oxygen mixed CO2 and water, and we talked about how there's 4 steps, of cellular respiration. First glycolysis, then the formation of acetyl-CoA, citric acid cycle, then oxidative phosphorylation. In this video, we are talking about glycolysis. So, glycolysis happens in the cytoplasm. As soon as glucose enters the cell, crosses the cell membrane, it starts to undergo glycolysis. So that's what we want to take a look at, Overall, it's just the idea is turning glucose into pyruvate, but we want to get into the nitty-gritty of how exactly that occurs. So let's take a look at the reactions. Now what I'm going to do here is I'm going to go over how every single little step in this metabolic pathway occurs and the general types of enzymes and the coenzymes or cofactors we may need for each step. The overall reaction, we have here is that, we're going to take glucose, which is our 6-carbon sugar here, and NAD+, so the oxidized coenzyme, and we're going to turn it into 2 pyruvates. Pyruvate. These 3-carbon sugars. So sometimes in biology books they show glucose as a 6-carbon sugar, and then pyruvate is 3-carbon sugars, which works fine if you draw little circles, but we're going to actually draw the molecules themselves. And it's important to note that in glycolysis, we require an NAD+, we make an NADH, or we make a pair of NADH and H pluses, and we make a pair of ATP molecules. So overall, we produce 2 ATPs. So let's take a look at our very first step, We're going to talk about converting glucose into glucose 6-phosphate. And so if you take a look at these molecules, the main thing that happens here is that glucose at the bottom does not have a phosphate, and now suddenly it does have a phosphate. So where did that phosphate come from? If you remember back to all the different cofactors and coenzymes we talked about, we said that one of the 2 jobs of ATP was to be hydrolyzed to provide energy to power an uphill reaction, an endothermic and endergonic reaction. And the other one was to add a phosphate onto organic molecules, which is what we see here. So in this case, it's ATP is the molecule that's helping us add that phosphate on. So I know that ATP is coming in, and when it donates that phosphate, it becomes ADP. One way to look at this here, is that you have the adenosine piece with the ribose and everything, and you can imagine it having 3 phosphates on it. And the idea is that one of those phosphates is going to get put right here onto glucose, and then it leaves as adenosine diphosphate, So now it only has 2 phosphates on it. So we've used up one of our ATP molecules to phosphorylate glucose to turn it into this glucose 6-phosphate. Now, what type of enzyme, We have enzymes that pretty much control almost all of the steps in glycolysis, all but one. If you look at this, since we're transferring a phosphate, it must be some type of kinase. And remember that our enzyme types, that we're just going with classes of enzymes. We're not going to talk about phosphoglucokinase or really specific names. We're just going to do general names like kinase, isomerase, oxidoreductase, hydratase, and hydrolase. We're just going to have those big classes. If you don't remember what those enzyme types are, go back and rewatch the end of that enzymes video that I had and take a look at that summary there. So we're able to turn glucose into glucose 6-phosphate by using a kinase enzyme and using up ATP to make ADP. In the next step, we convert it to the following molecule, the fructose 6-phosphate. And you take a look at this, I still have the phosphate at the bottom. I've got something going on here. I had an aldehyde at the top, and now I don't. And then I had just an alcohol on carbon 2, and now I have a ketone. So something's going on there. Maybe it's an oxidation, maybe it's a reduction. I should probably check to see if I gain hydrogens or lose hydrogens or gain oxygens or lose oxygens or something like that. So I'm going to just write the formulas here because this one doesn't seem as obvious to me. So it looks like I have 1, 2, 3, 4, 5, 6 carbons. So this is a C6. Hydrogens, I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I've got 11 hydrogens. I've got 1, 2, 3, 4, 5, 6, 7, 8, 9 oxygens and 1 phosphorus. And if I look over at the other molecule that it becomes, I have 1, 2, 3, 4, 5, 6 carbons. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 hydrogens. And I have 1, 2, 3, 4, 5, 6, 7, 8, 9 oxygens and a phosphorus. So these look like the same formula, right? Or in other words, these must be isomers. But they have the same formula. I didn't add or take away anything for this molecule. We just rearranged stuff. So that means I know that the enzyme must be an isomerase. Must be an isomerase because there was an isomerization that occurred. It must be that we just rearranged the atoms from one thing to another. So glucose 6-phosphate is just an isomer of fructose 6-phosphate. That's all that really happened here. Now if I take a look at the next step, right, so step 3 here, I see again that it looks like I have now a new phosphate up here. I didn't have a phosphate before, now I do, so it must be similar to that very first step, or in other words, I must have taken another ATP, turned it into an ADP, using some sort of kinase to catalyze that reaction, because again, kinases catalyze the transfer of phosphate, and here we transferred phosphate from ATP to ADP. Now in my reaction, right, we said that we're supposed to make 2 ATP, but so far we've used up 2 ATP. So we're actually 2 in the hole. We've lost 2 ATP so far in these first couple of steps. So hopefully, right, we'll see in the next few steps that we're going to start making ATP. So we're not done. We still got quite a few reactions. So let's move on to the next slide here. So we got to convert fructose 1,6-bisphosphate into something new. And if you look at that something new here, right, so I've got the bisphosphate. This reaction, I'm not going to ask you guys, I'm not going to hold you guys accountable for this reaction, but this is where we take our molecule and we actually cut it in half. So we go from that 6-carbon piece to 2 3-carbon pieces. Where if I label these carbons 1, 2, 3, 4, 5, and 6, this is carbon 1, 2, 3, and 4, and 5, and 6. So you've just cut it right in half is the idea. Again, I'm not going to ask you what this enzyme type is because in my class we haven't talked about what an aldol reaction is, but it's just a type of aldol reaction, and so this would be considered an aldolase. You just slap ace at the end of it, it's an aldolase. Now the idea here, what's pretty cool, is that instead of having 2 pathways here, right, where you take this dihydroxyacetone derivative and start reacting it also, what we do instead is we convert it to this. And so the idea of this glyceraldehyde, let me write that down. So this is a glyceraldehyde. It's a phosphoglyceraldehyde. But it's a glyceraldehyde derivative. And this is the dihydroxyacetone. Dihydroxyacetone. The idea is that we take the dihydroxyacetone and we convert it to glyceraldehyde so that as we move on through this synthetic pathway, all of this stuff happens twice. So what we're going to see is that we're going to get 2 glyceraldehydes, and then there'll be 2 of these, and everything's going to happen twice because effectively what's going on here is we're going to make 2 glyceraldehydes. We're going to make one of them out of carbons 1, 2, and 3, and one of them out of carbons 3, 4, and 5. So PO3 minus 2. Right, so this one up here basically is going to make a second glyceraldehyde. So we're going to end up with 2 glyceraldehydes at the end. We're going to take the top half, chop it into dihydroxyacetone, which is then going to convert into glyceraldehyde, and we're going to take the bottom half and convert it into a glyceraldehyde also. So we're going to end up with 2 glyceraldehydes, and that's going to just be carried on through this metabolic pathway. And if we take a look at the glyceraldehydes, right, I've got an interesting reaction that happens here. Looks like at the top I had 2 bonds to this carbon up top, 2 bonds to oxygen, and now I have 3 bonds to oxygen. So it looks like I've oxidized it, which means I now need to do something with an oxidoreductase. I need to have some sort of coenzyme also. So I know that I have an oxidoreductase because this is a redox reaction. Some sort of oxidoreductase. And in this case, since I'm doing a redox reaction with a carbonyl, right, not an alkene with a carbonyl, I know that NAD plus is probably the coenzyme that's being used here and not FAD. So remember that carbonyls are usually involve NAD and alkenes usually involve FAD. So it must be that this NAD plus is what's plucking off that hydrogen to make NADH. And then the other thing that's happening in this reaction that's not quite, is not super apparent, is that phosphate that's being added here actually doesn't come from ATP. It just comes from free phosphate, PO4 minus 3. And that free phosphate is going to give us another H plus is the idea. And again, this occurs twice. Oops, not 3. This is a 2. Excuse me. 2. And so this is an oxidation reduction reaction, right? We're going to oxidize that aldehyde up to a carboxylic acid derivative, and we've reduced our NAD plus to NADH because we've added hydrogens to it. And so this is where we actually use up 2 NAD pluses, right? We've used up 2 NAD pluses in this step because we have 2 glyceraldehydes, one from 1,2,3 and one from 3,4,5. And we're going to turn those both into this other compound, this this other compound. I'm not going to name all of these for you. And so we have 2 of these that now convert to this last compound, where it looks like the big difference here, right, is that there was a phosphate and now there is not a phosphate. And so again, we've transferred a phosphate. So it's the same enzyme, whether you transfer it on or off. And so it must be a kinase, some sort of kinase that is involved in this one, right? We're going to lose this phosphate. And what's interesting here, right, is we're going to lose the phosphate, but instead of putting it onto ATP, ATP's already got the 3 phosphates. It's not going to be 4 phosphates. There's no adenosine tetraphosphate or anything like that. We're actually going to put it on a DP. Whoops, excuse me. Let's get my colors straight here. We're going to put it on a DP, the diphosphate, to regenerate some ATP, right? So we're basically taking this phosphate and putting it onto ADP to make ATP. So we're going from the diphosphate to the triphosphate. It picked up that 3rd phosphate from this molecule here. And since, again, all of this is happening twice, at this point, we've now broken even with our ATP count, right? We used 2 ATP on that first slide, and now in this step right here, we've just remade 2 ATP, because again, we've done that. We've ran this twice with those glyceraldehydes. And you'll notice I got a little arrow here, so we got to talk about the next step. We got to keep on going here. So let's see what else. Let's finish this up. So I've got my I've got the molecule from the previous slide here. I've lost that phosphate. In this step, it looks like my phosphate just transfers from the last carbon to the second carbon, so from carbon 3 to 4. All I've done is move that phosphate around. Since that means I didn't add anything or remove anything, it must be that I just used an isomerase. If you took a look at the formula for both of these, you'd see that they have the same formula. It's just an isomerization reaction. I moved the phosphate from carbon 3 to carbon 2, that's it. And since it's an isomerase, I don't need to add anything, right? I didn't add hydrogens or take away hydrogens or add phosphates or anything like that. So I don't need to put anything coming in or leaving. No, I don't need any cofactors or coenzymes. Now in this second step, I kind of got a lot going on here, right? So I still got the phosphate on there, but now I've got this like double bond. I've got an alkene in there. Looks like my OH is gone down here. So I've got no more OH down there. Right, this OH is gone, and now I have an alkene here. So I might be thinking that like maybe I need FAD or something because I make an alkene show up. But again, if I'm not too sure, I should just always make that. I should always just figure out the formula. So for this compound right here, it looks like we have 1, 2, 3. So this is C3 H4O 1, 2, 3, 4, 5, 6 P. This compound is 1, 2, 3 C3 H2 1, 2, 3, 4, 5, 6. Sorry, 1, 2, 3, 4, 5, 6. Oh, this one was 7, excuse me. See if I can count. Right, 1, 2, 3, 4, 5, 6, 7. And this one was O6 and had 1 phosphorus. So if you look at the difference between these two, right, my hydrogens go from 4 to 2, and my oxygens go from 7 to 6. Or in other words, what I lost here was 2 hydrogens and an oxygen. I lost 2 hydrogens and an oxygen. Or in other words, I lost water. I lost H2O. And since I lost water, right, I know that this must be something catalyzed by some sort of hydratase. Because we said that was the enzyme class that's involved when you add water or remove water from a single molecule, right? It's different than hydrolysis. It's different than a hydrolase, where you're combining 2 separate carbon pieces or breaking up 2 carbon pieces. This is just within one thing. So we've just lost the elements of water. I don't need to put anything coming in. Nothing came in. It's just that water left. Water leaves this. So some sort of hydratase. Now in this last, or this penultimate step here, right, it looks like I had a phosphate here, and now I don't. And so again, a phosphate left. So now I'm thinking, again, a kinase, some sort of kinase. And again, if I'm going to put the phosphate on ADP, my low energy molecule, to make more of my ATP, my high energy molecule. And remember that since all of this stuff happens twice, now what we've done is we've netted, this last part is where we've netted that 2 ATP. So this happening twice is what gives us that last 2 ATP that we've formed, which is great. So glycolysis has a net production of ATP. Now this molecule here, this is an enol. I don't know how much we need to get into this, but the idea is that it's very, very unstable. Very, very unstable, and it's just going to spontaneously react, spontaneously transform or tautomerize to pyruvate. So spontaneously converts to pyruvate. Right, so there's no enzyme. I don't need anything here. You just leave this blank, this last arrow. There's no enzyme that catalyzes it. There's no cofactors that need it. It just undergoes what's called a tautomerization for those of you who've had some organic chemistry. And then boom, right, again, remember this all happens twice, so now we've made our 2 pyruvates, our C3O3H3-. And that's what glycolysis is, right? So that's all the nitty-gritty steps of converting, right, all of these reactant things into all of these product things. That's how we do it. That's step by step by step by step. And so for my students, I'm not expecting you to memorize this. I don't need you to memorize all of glycolysis. But what I do want you to be able to do is if I show you that some molecule turns into some other molecule, just one step, that you should be able to identify what type of enzyme, what class of enzyme, and you should be able to identify what cofactor or coenzyme you need. You know, do you need ATP? Do you need NAD+? Do you need FAD, etc.? And I'll show you some more examples in the following videos. If you have any questions or comments, please leave them down below. I will also post a video on running glycolysis aerobically or anaerobically. You'll notice that in this overall equation and in this pathway, I never wrote O2. We did not need O2 for any one of these steps. So technically, glycolysis doesn't involve oxygen directly. One of the reasons we treat it as being anaerobic. It's a little more complicated than that. Again, I'll post a video about glycolysis being driven either aerobically or anaerobically and when that occurs. Happy studying.